So we are going to talk about signal transduction. So if you don't already, go ahead and grab your notes out. We are hopefully going to finish up all of this content by tomorrow. That's my goal. And then next week we get to talk about mitosis, which is always super fun. Yes. Beautiful. Okay. So, we have talked about so far the types of external signals. So, the different ways that cells send signals to one another, cells talk to one another. And yesterday, we talked about the different ways that receptors actually get those signals, right? We talked about G proteins and how G protein coupled receptors work. We talked about receptor tyrosine kinases and how their job is to like phosphorylate tyrosine and how they work when they are in like two little dimers. And we talked about ligand gated ion channels, which when a ligand binds, open up and let ions through a membrane. So those were 3 different types of receptors. And when we talked about those receptors, we also, like at the end of it always ended in some sort of cellular response. But in order to get to that response, more often than not, there is some sort of intermediate pathway that has to happen in order to trigger the cell to actually have that response. So we'll talk about responses tomorrow. Today, we're talking about signal transduction, the pathway that signal takes after the receptor receives a signal, but before the response actually happens. So we talked a little bit about signal transduction pathways in like our overview. So we're going to kind of bring that back a little bit. So what is transduction? So I've said transduction probably a million times at this point. But we don't have a like actual technical definition for it. Transduction is a cascade of molecular interactions relaying signals from receptors to target molecules. So if we've got a receptor, it's going to have an interaction with another protein, which would have an interaction with something else, which has an interaction with something else, which eventually gets to our target. So these can be proteins, these can be messengers, different types of proteins. This is just what transduction is. It's like a game of telephone in your cells, right? One person says one thing, the next person repeats it, the next person repeats it in order to get it where it needs to go. Like an assembly line. People are trying to get heavy things over long distances, right? So it's this cascade, they like the word cascade, of interactions that will get our signal from point A to point B. That point B being our target molecule. That's the goal here. That's what we're talking about. How this like intermediate step happens. This is today. Receptors was yesterday. So, I'm on the wrong writing thing. Oopsies. Yes, there we go. So, signal transduction pathways are usually, my spaces got messed up in here, the majorly like dependent on protein to protein interactions. So, my space bar was a little bit behind where my words were. All right, the major theme of cell to cell communication. Um, So, this is usually how these cells communicate with one another, which is how cell signaling works through protein to protein interactions. This is typically done by protein phosphorylation and dephosphorylation. Big, strong words that just mean adding and subtracting phosphate groups. Those little phosphate groups that we learned about like the first day of school, not first, but like third or fourth. So, protein to protein interactions are like our overarching theme for signal transduction pathways. It's typically done through phosphorylation and dephosphorylation. And as we talked about yesterday, when we talked about receptor tyrosine kinases, kinases are things that do the phosphorylation and dephosphorylation. Kinases are those special enzymes. ACE. Got that ACE in there. Remember, ACE means enzyme. Kinases, they are what's doing this phosphorylation. They're kind of leading the pack with these protein to protein interactions, as they will take a phosphate group off of one protein and add it to another. So these kinases are like super big players in these transduction pathways because their job is to move stuff along. They are like doing a lot of the heavy lifting in these little cellular games of telephone. Questions so far? I feel like I'm talking really fast. All right. So this often leads to a phosphorylation cascade. I said they like the word cascade a lot. So a phosphorylation cascade is a series of different proteins that are phosphorylated that will each kind of add a phosphate group to the next protein in line. So a phosphorylation cascade. So we've got a protein, another protein, another protein, another protein. A phosphorylation cascade is taking a phosphate group from this protein and having a kinase move it to another protein. So now it has the phosphate group, and a kinase will then phosphorylate another protein and dephosphorylate the one it took it from. So it's actually removing these and taking this and moving it to the next one. This is a phosphorylation cascade, moving these phosphates down a path through the cell. You mind my weird shorthand. These big ones are proteins because I have a line on it. These are phosphate groups. So protein, phosphate. This is our phosphorylation cascade. We talked about kinases being kind of our driving force here. They are what is moving these proteins or adding these phosphate groups to new proteins. But there's another kind of player at hand here doing the dephosphorylation, right? So there are these things called protein phosphatases. Phosphatases, ACEs. ACE means enzyme. These protein phosphatases, they rapidly remove. Ooh, okay, spell check didn't work on this one. Rapidly remove phosphate groups from proteins. They do the dephosphorylation. They do the dephosphorylation. 1 second, I gotta check something. There we go. So we've got our kinases, which are adding phosphate groups, and we've got our phosphatases removing phosphate groups. So it's actually our protein with our phosphate group, and we have a phosphatase which is going to remove that phosphate group. And then we have a kinase, which is going to add that phosphate group to another protein. So phosphatases remove phosphate groups. Kinases add. The word rapidly in here is also kind of important. These signals get received and transmitted very, very, very quickly. This is not a slow process. Cells are very fast to responding to big events like this. So these are protein phosphatases. They're another kind of big idea here. They're another player in these phosphorylation cascades. So we are going to draw out like a skeleton, like general idea of what a phosphorylation cascade is actually going to look like. So this is our signal transduction pathway skeleton. We're going to start outside the cell. So we're going to draw our cell membrane. This is outside the cell. This is inside the cell. And we've got our receptor protein. This right here. This is our receptor. This can be any of the ones that we talked about yesterday. This could be a gated ion channel. This could be a G protein. This could be a receptor tyrosine kinase. That's my the one I'm forgetting. Thank you very much. Huh? Ligand gated ion channel. Did I push those two words together? Oops. Ligand gated ion channel. It's like one of the three. So that receptor, when it gets our signal, it is going to activate a relay molecule. So we've got an activated relay molecule. This could be one of the proteins that got activated by the phosphorylated tyrosine on those receptor tyrosine kinases. This could be that enzyme that the G protein activated at the end of G protein signaling. We've got a relay molecule. And that is going to activate a protein kinase. So we've got our inactive protein kinase, and this molecule is going to activate it. And we are now going to have an active protein kinase. All right. That active protein kinase is actually going to use an ATP, because ATP is like what we're going to do, what uses cellular work, and it's going to take that phosphate and activate a second protein kinase. So we're actually going to call this protein kinase 1, and we're actually going to have another inactive protein kinase 2. And this activity right here, this phosphorylation, is actually going to activate protein kinase 2 because now it's got a phosphate group on it. this will then activate our cellular response. But it is just as important to turn signals off as it is to turn them on. So, what's going to happen is that this molecule, through a protein phosphatase, is actually going to release that phosphate group and go back to being inactive. So protein phosphatase is going to remove this phosphate group and send it back to being inactive, where it will remain until it gets activated again. Like I said, this is just a kind of skeleton idea of what this will look like, because some signaling transduction pathways have like 4 or 5 of these systems in a row. Sometimes there's fewer steps. This is just like our general idea. Questions here? All right. So, not every molecule in a signal transduction pathway is a protein. There are other molecules that are not proteins that are involved in these signal transduction pathways, and we call them second messengers. These are small, non-protein, water-soluble molecules or ions that will spread throughout a cell via diffusion and usually participate in interactions guided by G protein coupled receptors. Remember, these are G protein coupled receptors and receptor tyrosine kinases. Just in case you don't know what those stand for. So they are small, they are water soluble, and they can diffuse through the cell. This is super important because proteins typically bulky, clunky, they don't move very fast. Diffusion, super quick process. Super quick process. So by having these systems in place, using these second messengers, we are rapidly picking up the pace. Not that our other systems were slow, they were still fast, but diffusion happens like that. So these second messengers can like go faster, go farther, send signals in a more efficient way than our protein intermediates, our kinases, our phosphatases. Second messengers are quick. These are typically in pathways by G protein coupled receptors and receptor tyrosine kinases, which means our ligand gated ion channels aren't really a part of this. They don't typically, they use ions, obviously, because ion channels, but those are kind of more direct players than second messengers. Questions here? I've got 2 very fast second messengers to talk to y'all about. They are cyclic AMP and calcium ions. So, cyclic AMP and calcium ions. Those are the 2 we're going to talk about. Cyclic AMP, also called CAMP. So you'll hear CAMP a lot. But the C is lowercase. Also, AMP, not a new molecule. it's ATP with 1-phosphate, right? So ATP, adenosine triphosphate, ADP, adenosine diphosphate, AMP, adenosine monophosphate. So same stuff we've been working with. It's cyclic because it's a little circle. So cyclic AMP. And then calcium ions. Calcium ions. So cyclic AMP. So what happens with CAMP is that an enzyme embedded in the plasma membrane will take ATP and convert it into AMP or CAMP, right? In response to a signal. So one of the G protein coupled receptors, one of those enzymes it activates, could be this adenyl cyclase. You don't need to know that. I just kind of want to throw that out there. Could be this adenyl cyclase, and it rapidly just starts cranking ATP into CAMP. Now, usually the cellular concentration, so the concentration of CAMP in a cell, is relatively low. This enzyme can multiply the concentration of CAMP, the normal concentration of CAMP in a cell, by over 20 times. Almost like in seconds. In seconds, can multiply the concentration of cyclic AMP by 20. This rapidly broadcasts this signal through the cytoplasm. As that concentration increases, right? The concentration increases, that diffuses throughout the whole cell. And by diffusing out through the whole cell, we get that message where it needs to go fast. And there are a number of pathways cyclic AMP can take. So we activate this enzyme, boom, it just starts cranking ATP into a cyclic AMP, multiplies that concentration by a lot. Now there's an absolute boatload of cyclic AMP chilling in the cytoplasm, which naturally, because it's small and water soluble, kind of diffuses where it needs to go. Like if you dropped a drop of food coloring in a cup of water, right? It kind of gets everywhere relatively quickly. That's what's happening with cyclic AMP. We're churning it out, we're pouring it into the cell, it kind of diffuses everywhere it needs to go. It's a second messenger. Super common in a lot of cells to have cyclic AMP be their like main second messenger. Questions about CAMP? If you ever find yourself in a molecular biology course or a developmental biology course, you know, talk about CAMP a lot. All right, and then calcium ions. Calcium ions are one of the most, if not the most common second messenger. We use a lot of calcium ions. Things like neurotransmitters and growth factors and hormone pathways use calcium ions. So the little messages being shot between neurons in your brain, getting you doing what you need to be doing, like your brain function, depends on calcium ions. The increase in these calcium ions, like a sudden increase in calcium ions, can trigger a lot of different pathways. A lot of different pathways. questions here. Calcium, super important. So we talked about what made CAMP, right? That enzyme that turned ATP into a cyclic AMP. We don't make calcium ions. Like our bodies can't make calcium. So what happens is we store it until we need to use it. So normally the concentration inside the cell, like the concentration of calcium ions inside the cell, is much lower than outside of the cell. So if you've got like a cell here, we've got a very, very low calcium ion concentration and a very, very high calcium ion concentration outside the cell. So super low inside the cell, much higher outside. So this is because we store a lot of calcium ions in the endoplasmic reticulum. When we talked about cell organelles, we talked about all of those different functions. We talked about the smooth endoplasmic reticulum, right? Smooth endoplasmic reticulum is like my favorite organelle. And I feel like it gets overlooked a lot because it's not doing the protein stuff like the rough endoplasmic reticulum. But the smooth ER, it stores all of our calcium ions and plays a super big role in cell signaling. So we take all of the calcium ions inside the cell and we store them in the smooth endoplasmic reticulum. That way we keep the concentration of calcium ions low inside the cytoplasm. This is on purpose. We do this on purpose because it is the release of those calcium ions that triggers our signals. So we've got all of our calcium ions inside our cell. This is the last slide. In our response to our signaling pathways, so after our signal transduction pathways, can trigger the smooth endoplasmic reticulum to release a bunch of calcium into the cytosol. And because the normal concentrations inside the cytosol are super low, any change is a big deal. So we can have our outside receptor get our signal. Our signal can get passed from protein kinase to protein phosphatase to our second messengers and hit the ER. And the ER can be like, oh, my time to shine. Shove all of those calcium ions outside of the smooth endoplasmic reticulum into the cytosol. The cytosol suddenly has a much higher concentration of calcium ions. And because of that, we've got signals. The sudden release of calcium ions in your nerve cells is super, super important to sending nerve impulses. Questions about signal transduction or second messengers? Yes, no, maybe so. All right. Well, that's what I have for you today. There is an exit ticket on Canvas. If you could fill that out for me, please. It is 4 questions. 2 of them cover yesterday's content, and 2 of them cover today's content. So if you could knock that out for me, you have about 5 minutes left. If you don't get it done, that's fine. Just get it in before the 10th if you could. Sooner better than later, but that's what I've got for you.